I've had a transcription and translation request, which makes me so happy because I love it. What we're talking about here is transcription, and this takes place in the nucleus of a cell. So we know that this is a eukaryotic cell. If you're ever asked about a nucleus and how you know what it is, it's actually always a 3 marker, and there are 3 things that you need to say. One is that it is a double membrane bound organelle. So I'm just doing these here. These are the nuclear pores, which is where mRNA can leave. So the first thing that you say is that it is a double membrane bound organelle. The second, you talk about the presence of a nucleolus. So that is in the middle, like this dense bit of DNA, and that will code for producing ribosomes out in the cytoplasm and on the RER outside. And the third thing, I think you say, nuclear pores, presence of nuclear pores. Okay, so we've got our DNA here. That has to split, so it separates. When you talk about transcription, you don't actually need to talk really about DNA helicase, which is the thing that the enzyme that does the unzipping. By unzipping, we mean the breaking of these bonds. Just because if it's a transcription and translation question, there's so much to it, you don't necessarily get extra marks for that finer detail. But if you had a longer exam question purely on just transcription, then you would talk about the fact that DNA helicase is the enzyme that will break these bonds. So a bit of a quick recap. We've obviously got our sugar phosphate backbone in the DNA. This is made up of the ribose sugar, the phosphate, ribose sugar, phosphate, and the bonds down the side, we call those phosphodiester. So that's just a combination of ester bond, phosphate, ester bond, and that's what makes up our backbone. Here we know we have got a combination of bases in a different order, and they match by complementary base pairing. Between those, you've got our hydrogen bonds. There are 2 hydrogen bonds between A and T. Between G and C, there are 3. I always think at GC, the GC Gemma Collins, bigger, bolder, better. We've got 3 bonds there, but only 2 between the A and T. So back to this part, transcription takes place, and what transcription is taking a copy of a gene, so these base pairs in, or a base sequence, in order, and you're turning that into an mRNA molecule that is able to leave into the cytoplasm so that we can convert that sequence into a protein. We know that that is called pre-mRNA, so you did that in year 13. Pre-mRNA will be created, and then we have spliceosomes, which will splice out the introns, which are the non-coding regions, which we believe are to do with switching on and off the different genes, and then the exons, which are kept in, and we know that those can be combined in a different combination to make different proteins. So you could have exon 1, exon 4, exon 5, exon 6, and that might give you one type of protein. And from the same strand of gene, you could have exon 1, 2, 8, and that would give you a second type. So helicase is the thing that does the unzipping. The next thing that we've got here is our RNA polymerase. So don't get confused with DNA polymerase. DNA polymerase is when we are creating a second strand of DNA for mitosis to take place. So when the cell divides, we